this?
Yeah.
Yeah.
Yes.
Great. First of all, I would like to pronounce your name so that we can pronounce correctly as well.
Yes, my name is Michele Ceccarelli.
Ceccarelli.
Yes.
Right.
Yeah.
And where are you from?
I'm from Italy.
And where are you talking from now?
I'm talking from Qatar. I am also working in Qatar at the Qatar Computing Research Institute, which is a part of university from Qatar, Hamad bin Khalifa University. I'm also associate professor at the University of Sannio in Italy, Benevento.
And Sannio is a city?
No, Benevento is the city, Sannio is the area, the region.
Oh, okay. Great. I'm sorry for the ignorance.
It's fine.
So let's start the interview itself. How did you get involved in the first time with studies about glioma?
My group, I'm a computer scientist, and my group develops methods and algorithms to analyze genomic data. We have developed some tools to model the cell as a network of molecular entities that interact each other. We also developed some tools to detect some genomic alterations like mutations and other kind of alteration in cancer genomes. And so I started in 2011 a collaboration with a group in Columbia University where I participate to the discovery of a novel molecular alteration, which is called fusion, that affect a small group but a significant group of patients with glioblastoma, which is the most aggressive form of glioma. And I was involved in this project by the group of, which is led by the group of Professor Iavarone from Columbia University, Dr. Berrak from MD Anderson, and Professor Yutan Yushmer from University of São Paulo. And we joined to this big effort to try to characterize better gliomas.
Great. And as you work with other types of cancer, what do you think are the particularities about glioma that are different from other types of cancer?
Because there are very few common molecular alterations and very few known drivers, and which can be also some therapeutic targets. And also, gliomas are represent 80% of all brain tumors and are of malignant brain tumors, and so are particularly aggressive, and some types have no cure. So this is one of the main challenges for scientific community.
You said that in 2011 you participated in the first study with gliomas and you discovered a particular molecular subject about it. What is the name exactly?
It's gene fusion. Not me. I participate to this group, to this discovery, and the main discovery was from the group of Columbia University, and the name is gene fusion. It's a particular alteration that indeed could be even targeted by therapies.
Do you remember which genes are fusioned?
Yeah, gene FGFR and TACC, TACC. This is the particular fusion that we described for the first time, and this was my first work in gliomas.
And how did you get involved with this actual project that's going to be published by Cell?
Oh, yeah, because we have developed some tools and some algorithms that can be useful to analyze this kind of data, in particular gene networks and analysis of gene signatures, then knowing people that led the study, like Professor Iavarone and others, I got involved, and I was excited and tried to contribute as much as I could to this project.
So if your lab also does do an analysis of the data, what's the difference between your lab and Houtem Nushmer lab?
Oh, yeah. It's the difference that we try to look at this disease by using several genomics platforms and several kinds of data. So the main point was to try to integrate different kinds of analysis in such a way that we can observe this phenomenon at different levels, looking at the mutation events, copy number events, methylation events, and gene expression events and protein expression events. And on our part, we made most of the analysis in gene expression, genetic signature, and deriving groups of gene expression. And on the other part, the group of Professor Newton at UCP, they made a strong contribution on the epigenomic landscape, to characterizing the methylation status of this kind of samples.
You, in the article, you say that you found or listed 75 genes correlated to gliomas. What's the importance of finding these genes and correlated to the glioma?
Yes, these are the frequently mutated genes in gliomas. So these are the most significant alterations that we observe at mutation level. At one of the events that we observe that are commonly observed in gliomas. But the pattern of mutation can be different between groups that are created by looking also to other events like methylation and expression. So these 75 genes then have different pattern of alteration among the various groups, different percentages and so forth.
And is it important to diagnose correctly the glioma?
Oh, yes. The diagnosis is now based on some histological parameters. And the main point in this project was to have a way to better characterize gliomas at the molecular level, because there is some discordance between diagnosis using just histological and histoclinical parameters. So molecular parameters are less sensitive with respect to the subjectivity of pathologists. So the join together histology, histological, histoclinical parameters and molecular parameters it's a way to have a better diagnosis of gliomas, because there is between 25 and 50% discordance in diagnosis, and treatment is based on diagnosis. So the diagnosis is very, very, very important. These were the main clinical requirement and the need for this study.
Do you think that treatment can change if you make these two analyses together, the histological one and the molecular one?
It will mainly depend on the acceptance of clinicians of these studies, but some of the things that we observed is that there are some subgroups of patients that could spare aggressive treatment, because they have a pattern of a molecular profile that is less aggressive than seems to be less aggressive than other in the same subgroup. So the group that we defined in this paper then can lead to a different of treatments among the patients, but mainly because there is a better diagnostic tool. Molecular information can be a better diagnostic tool.
Great. If you are the first author, I would like to ask you, how did you conceive this study? How did you think to put these information together?
I am the first author together at the same with the same responsibility, let's say, with other three people: Floris Bartel from MD Anderson, and Tatiana Malta and Thais Sabato from USP. So we are all first authors. Just for maybe alphabetic order first. The main point here was to try to integrate and to get the most comprehensive data set with integrating different molecular platforms to try to come out with these to try to come up with a better characterization of this set of gliomas. We used about 1,100, more than 1,100 samples, and the groups that we were defined that were then validated also in other data sets.
And this kind of study has already been made to other types of cancer, this kind of comprehensive study?
Yes, there are some pan-cancer studies, but this is a pan-glioma study. It means that we are focused on all the all the data that we can share, that we can collect about the gliomas. And this is the first study on the type of panglioma analysis. And the most interesting thing is that we used all public data, all data that can be used by other groups. So all the data is available for research and analysis to other groups. And this is the main feature, the main characteristic of this study, to share as much as possible data with other groups and to come out with novel discoveries.
I have read some articles about breast cancer, for instance, that have already this kind of analysis for breast cancer, lung cancer, and also prostate cancer. Do you think glioma has been in focus later than the other types of cancer?
No, glioma, no. Glioma is being so aggressive, it receives a lot of attention from by the scientific community. And this is not the first paper about gliomas. The TCGA published two papers on the glioblastoma, which is one of the tumors that we are analyzing, and another main important paper about lower grade gliomas. And so this is not the only or the first study of this kind, but maybe this can be considered the most comprehensive.
Going back to the study now, which were the questions you wanted to answer with this study?
The main question was very simple. We were looking for a better molecular characterization of gliomas, a better stratification of patient of glioma patient on the basis, despite the clinical histological diagnosis. This was the question. Try to have a better characterization and a better stratification of gliomas.
And what were the results of it?
The main result is that we can have several groups of several subtypes of gliomas, and the most important event is the methylation, and the groups are driven by methylation. This is one of the main results of this study.
What does it mean that they are driven by methylation?
That there is some signatures that can be measured and that can be analyzed and that define the groups that we are describing. And this means that once that you have these groups, then there are a lot of further studies that should be done to analyze group by group and to look at the molecular alterations within each group.
If you think of the histological classification and this one proposed now, do you think this one is more accurate than the histological one, has more subgroups? What are the differences mainly?
The difference is that they overlap each other in some way. And this classification is based just on the molecular information, no other kind of information. And as I told you, it is not subjective in some way. So it will not change between different clinicians.
So you have an objective parameter.
Yeah, this is correct.
Great. I read a lot about the classifications, and I think I could, I don't know if it's correct, you tell me if it's correct or not. I think I could see two very different groups that were not in the histological classification. That is the LGG, more aggressive, and the glioblastoma, less aggressive than the
glioblastoma is more aggressive.
Yes, but between the glioblastomas that are the most aggressive type of cancer, I found a subgroup in your article that means it's not that aggressive. And between the LGG types of cancer, I found that should be less severe. I found a group that is more aggressive. Is it the main difference your results show?
By using a so large data set, these numbers of patients, then we are able to give significance even to small subgroup. And so this is the advantage of using large data and observe many patients. So we can have some better characterization of these groups. And also what we observed was that there is some overlap between the two types of tumors, glioblastoma and lower grade glioma, which is some subgroup that goes have an outcome similar to lower grade glioma and vice versa.
This could not be found in the histological analysis, right?
Not, and this not be found without mining so many samples.
Do you think that the treatment for these patients that are diagnostic both by histological and molecular analysis, is the treatment going to be better for them? Do you think more precise?
I think that if you have better diagnosis, then the treatment can be better. So this was the main aim of this study.
And can you give an example? For instance, if you have a glioblastoma that is great for, but it's less aggressive.
We described a subgroup which is called PA-like that maybe is less aggressive than other. So some treatment in this case can be avoided. And so this is why this study is important, to try to better and to have some guide to better treatments.
When you conceived the study, you articulated several groups around the world. How did you do it, and how is it to work with groups that are so far away from each other?
It was a long run because we met every 2 weeks for more than 1 year with conference calls, and each group the participation to these groups is completely open. If you have something to say or some study or some analysis that you have done and you want to present it to the group, every research group is open and can have access and can participate and can present his result to the whole analysis working group. This is one of the most interesting part of this collaborative project. that every group in the world can participate and present its own result. Then when we observe that some results are particularly interesting for the project, then we go further and try to collaborate. And this is why we now started a strong collaboration between my group and a group of Professor Yutan, trying also to go forward with several other questions that arise once that we have this kind of classification.
Can you give an example, for instance, that someone gave some data that thought it was not important, but other group saw it and, wow, this is relevant. Can you give an example of this so that we can have it more?
No, I think that every kind of data, all the data are interesting. It depends on the way that which you observe this kind of data. For example, we observed one of the groups that we described and we discovered was originally considered as a single group. Then looking at, and this was the main one of the contribution of the group of Professor Yudan, looking at this kind of data set, then this group can be split in two different outcomes in two different subgroups, and this means that you have you can have a better knowledge of what is going on at molecular level in the various clusters that we observe.
Are you talking about the GSIMP high and GSIMP low specifically?
Yeah, correct. You are very well informed.
I read the article, yeah. Was it a surprise for you? Was there any surprise in the results?
The surprising result was that something that was considered different, indeed it was not one part, and something that was not expected. This GSIMP low was not expected, and so this something that every discovery means that you observe something surprising, something which is not known and which is not expected.
So when you started the work, you didn't expect to make this difference between the two groups, and it just came through the analysis.
Yeah, correct.
Oh, that's really, really interesting, because we always think that the scientists have already something in mind that they want to prove, and now they're going to receive the data and see something else from it. How does science work in this way?
We are doing a data science. In this case, we are doing a data science. We try to come out with some hypothesis from the data, not from a theory behind. It's a different way of observing and performing research in terms of data science, trying to come out with some hypothesis from observing and mining data.
So you didn't have a hypothesis based only on theory?
No, not in this case. The result came from the analysis, but we validated the result in other data sets. So indeed, it is not just a phenomenon that we observe in our data, but also in other studies.
So I'd like you to explain it better, because
study each other. Okay?
I'd like you to explain it with more details, because people are not used to see a robust, great result that has checked itself in other bases. How did you do this process? What do you want to look at other data from other consortiums or things?
It's quite simple. You discover something, signature, some gene which is expressed or has some specific characteristic in a group which have a specific clinical outcome. and from the patients that you observe. Then you try to validate. You make the discovery from, you use some information from the discovery, then the discovery gives you just an hypothesis that you try to validate, or in wet lab, or using other data set that you use for the discovery. So this is a way of validation. This is the idea of validation.
And you did this in this work, right?
It was done. It was done in this work. The groups were then validated in other data sets. So from discovery and validation.
And you didn't use only the TCGA data to do it?
Also, the discovery was done with TCGA data, and validation was done with other data. See? So it's a confirmation of the discoveries.
Does it make you more convinced of the results?
Yeah, this of course makes you more confident about the presence of this group, or the things that you are describing.
We are talking with Huten also about the translational medicine and the importance of the data coming from the hospital, then going to the labs, the wet labs, then the dry labs and everything. Do you think in this particular case, does the study come from a clinical problem that you wanted to solve?
The clinical problem is the diagnosis in this case, a better diagnostic tool. This is the main. That can also be used in clinical practice. That when we publish the signatures and which are the features that drive and that characterize the various group, then they can also use it in practice for diagnosis.
Do you think on daily basis, patients that come to a hospital can be diagnosed by this new formula you proposed?
I think that this will be used together with other classical approaches. So it will not replace completely, of course, other diagnostic tools, but it will be used in conjunction to actual diagnostic tools.
And do you think it will, is it going to be accessible to people around the world? Or
yes. All the results and all the data and all the findings are public, available, and distributed worldwide on the CGA portal. This is one of the features of these studies, this kind of study.
I'm thinking about the access to the exams that has to be performed to have a molecular diagnosis. Is this access available to people around the world?
It depends on some exams with novel sequencing tools and the molecular tools. The molecular diagnosis is becoming even cheaper and accessible worldwide.
Would it have to be just one more exam, or is it a complex analysis that requires several arrays, several exams? How do you see this methylation parameter?
No. In the case of methylation, it will be just one analysis, and with the specific chips that are relatively accessible and quite cheap, not so expensive. I cannot predict the exact cost, but it's not really so expensive.
Is it possible to think in the future about a diagnosis kit that's going to be provided to hospitals and everywhere so that this diagnosis can be easier?
Yes, this is one of the product of this study, is the molecular signatures that define the groups, and the molecular signatures are then used to do the diagnosis.
Great. Just 1 second. Oh, yes. Was University of São Paulo participants relevant to this work?
Oh, yes. The University of São Paulo made a great contribution, because they had the responsibility of the epigenomic landscape of gliomas, and this resulted in the main finding described in this paper. And this was done by the group of Professor Houghton and his collaborator Tatiana Malta and Thais Sabato are the main authors of the first authors of the paper. I also had the opportunity to visit the University of São Paulo, the group of Professor Houghton. It was a great experience, both from a professional point of view and personal point of view. And I think that we will continue to collaborate with other on the subject for other studies.
So do you think this study can have a another study vinculed, how do you intend to continue this study then?
Yes, as every discovery in science, when you answer to some question, that some other questions arise. In particular, now what we want to do is try to characterize and look at the biology of each specific group, so that we have this group defined, and we can look at the difference between one group and another, which is the biology that drives, which are the main genetic drivers and possible therapeutic targets for each group. And one of the study that we are trying to do is to describe or to obtain what we call the master regulators of the groups, in terms of, this means that the genes that drive and control all the signatures, the pattern of expression and methylation that we observe between groups. So now the focus will be on each group one by one to better understand their biology.
Just to finish, can you describe in a simple way the 7 groups you are proposing in the molecular classification?
Yeah, in a simple way, we have 2 main groups, macro groups. One is IDH mutant group, and one is the IDH wild type group. Then, within the IDH mutant group, we observe 3 main subgroups. One is called GSIM low, which is characterized by worst outcome, and another is GSIM high, and another one is GSIM codel. All these 3 groups in the IDH mutant macro group have a specific methylation profile that distinguish the groups each other. Then,
in general, this microgroup is associated with a better prognosis or a worse one.
So this in general with a better prognosis, but the GSIM low group that has a worse prognosis. And the other macro group is the IDH wild type, which is divided in 4 other groups. One is called classic-like, mesenchymal-like, PA-like, and GBM, LGM6 GBM-like. And they have a very a worse survival, a worse outcome, but the PA-like, which has a better survival.
And in general, this group is associated with what? The IDH wild type.
So the IDH wild type are associated with different molecular alterations, the amplification of a gene called the EGFR, and amplification of deletion of part of chromosome and amplification of other part of chromosome. And in particular, among this group, the PA-like is the group that have less molecular, macroscopic molecular alteration in terms of what is called copy number.
It would say it has a better prognosis.
Yeah, definitely.
But in general, this group would be a worse prognosis than IDH mutant, or a better prognosis than an IDH mutant?
Worse.
Worse.
Yeah, worse prognosis.
So we can say that glioblastomas that are currently being associated with a worse prognosis, most of them would be in this group, right?
Yeah, most of them, but the GSIMP, some of the GSIMP that go in the first macro group, and they go within this group.
And the low-grade gliomas would be...
Glioma also, the IDH wild-type lower-grade gliomas, they also go within this group. And they are further subdivided in mesenchymal, just like the glioblastoma, with the exception of this group, PA-like, which is lower-grade glioma, IDH wild-type, that is a specific methylation profile and better prognosis.
Besides the better diagnosis, did you find anything that might suggest a target for treatments, for future treatments?
I think that this will be the next step of the study, understanding the biology of the groups and the identification of the targets.
Great. Thank you. Do you want to add something else, something that happened was, I know, amazing, and you want to share with us?
Yeah, the amazing was this collaboration that we established. We are so far, but we were able to integrate each other, our group. We knew each other, and we had a great personal experience in terms of collaboration and getting in touch with scientists from all over the world. And this was amazing and interesting. And this one of the things that stimulate research is also the possibility to collaborate with other people.
Just to finish, I'm sorry, just to finish twice, but do you have anything that might not be closed yet, and do you think that you need more data to validate it? Do you think that there is any issue in this paper that should be looked at, not as a definite conclusion, but something to be checked?
As I told you, every study has a conclusion related to the study, but also opens novel perspective for the future. As always, we arrived at a good classification that we rely, but now we have to better understand the reason of this classification. So every step in science is always made with small steps, and each step creates other questions.
Thank you very much, Dr. Michele, right? Michele. Thank you very much, Dr. Michele, and I hope we can talk soon about other discoveries you're going to make.
Okay, thank you.
Bye-bye.
Bye-bye.
It was a really pleasure to talk to you. Okay.